Good afternoon. My name is Nora Speicher, and I'm going to talk about unsupervised data integration. The method we are using for this is regularized multiple kernel learning, and we apply it to several cancer data sets in order to identify cancer subtypes. What happens in cancer cells is that a lot of aberrations are accumulated, and these aberrations happen on different levels of the cell. So, for example, at the genomic level, like mutations, duplications, and deletions, but also at other levels, the epigenomic level, the transcriptional and post-transcriptional level, and so on. Because of these diverse malignancies in cancer cells, there is no single data type that can capture all the subtleties of a tumor. This is why large-scale projects like the Cancer Genome Atlas accumulate this multidimensional data, where multidimensional data contains, for every patient, measurements of various molecular features. For example, you can see here we have mutation data, copy number, gene expression, DNA methylation data, and so on. Once we have this data, we of course want to analyze it, want to gain insights from it, and one first step of doing so can be to integrate it by dimensionality reduction. And here you see, a two-dimensional plot of cancer patients. Every gray dot is a cancer patient, and now the distances between these patients reflect the integrative similarities. So integrative means that they are based not on one individual data type, but on all the input data types that were available. So from this representation, one can move on and do follow-up analysis, for example, clustering in order to identify integrative subtypes. And we're interested in these integrative subtypes because we want to understand how cancer patients having the same cancer type are still different from each other, and what implications does that have for the treatment. This is why it also affects personalized therapy. So now we have the data, but how can we analyze it? And there are several methods doing so. One of them is iCluster. It's based on a Gaussian latent variable model. And as you can see in this picture, based on the different latent variables that are learned, one can represent the data and cluster the data in order to identify cancer subtypes. However, this method is computationally very expensive, and because every data type comes with a lot of different features, like DNA methylation sites or genes, one needs to do a feature pre-selection, and this pre-processing step, of course, strongly influences the final result that we obtain. Another method is similarity network fusion. As you can see in the picture, you have different input data types. From each input data type, you generate one patient similarity network, and these similarity networks are iteratively made more similar to each other until you end up with an integrative patient similarity network. In this method, one important step is the initial choice of the similarities that are derived from our data types, because from this we get our initial patient similarity networks, and this choice will also strongly influence the final result that we get. So we propose a method based on multiple kernel learning. This means that each data type that we have is first represented as a kernel matrix. And here, the nice thing about multiple kernel learning is that we don't have to know the good kernel parameters or kernel function, but instead we can also represent every data type using several kernel matrices, and then the method will automatically learn kernel weights that upweight important kernels and downweight kernels that are maybe more noisy or contain not so much information. Using these different kernel matrices, we do data integration using dimensionality reduction. And this serves to remove the noise that might be in the data. One can also use it to visualize the data in 2 or 3 dimensions. and do follow-up analysis like clustering of the data points, and each data point represents a cancer patient, such that we can come up with clusters of cancer patients, and then every cluster is a potential cancer subtype. Now let's have a look at the main method here. Multiple kernel learning for dimensionality reduction is a very general framework that allows the implementation of a set of different dimensionality reduction schemes, and it's based on the graph embedding framework. So here you see the optimization problem, and the objective function is the graph preserving criterion. This graph preserving criterion depends on the weight matrix W, and this weight matrix determines which dimensionality reduction scheme is implemented. So we decided to choose a local method, locality preserving projections. This is based on the k-nearest neighborhood of each sample, and so the weight matrix W just represents the k-nearest neighbor graph of our data. Using this graph preserving criterion, we can learn two parameters, the sample coefficient matrix A and the kernel weight vector beta. So the kernel weight vector tells us how important each kernel matrix is, and the sample coefficient matrix then projects our integrated kernel matrices into a lower dimensional subspace, such that we can project every patient into this lower dimensional subspace. When we implemented this, we realized that in certain scenarios, namely when we have a lot of different kernel matrices or a small number of cancer patients or samples, we see instabilities in our results, and this is why we added another constraint that regularizes the kernel weight vector beta in order to avoid these instabilities in that scenario. The method has then two parameters that have to be set. One is the number of dimensions that we want to project our data into, and the other one is k, the number of nearest neighbors for our local dimensionality reduction scheme. We applied this methodology to 5 different cancer data sets, brain tumor, which is our largest data set, but we also have breast, lung, kidney, and colon cancer. And for each of these cancer types, we have 3 different data sources, namely gene expression, DNA methylation, and microRNA expression. From the available data, we generated 2 different scenarios, the 3K, which serves as a baseline, and the 15K scenario. Both are based exactly on the same input data, but for the 3K scenario, we generate 1 kernel matrix per data type. For the 15K, we generate 5 different kernel matrices per data type, and this simply serves to try to understand if the method is able to do the weighting correctly. So for the 15K scenario, we also have kernel matrices that contain mainly noise, which should then be downweighted and not influence the final result strongly. The dimensionality reduction and clustering step is the same in both scenarios. So let's look at the results. We first did a survival analysis. So for each cancer type, we identified our potential subtypes, so our clusters, and we figured out if there are significant differences in survival times between these clusters. And here you can see the first column is the similarity network fusion approach that I introduced in the beginning. Then we have our 3K and our 15K scenario. And to represent it a little bit more clearly, I just show you the median here. We can see that the 3K scenario, our baseline, yields results that are comparable to the similarity network fusion approach. And what is important for us here, also the 15K scenario yields significant results. Also, we used partly kernel matrices that contain a lot of noise instead of information. So, as I said, we introduced this additional constraint to increase the robustness of the approach. And here you see the comparison of the robustness between the non-regularized approach and the regularized one in red. And since these instabilities occur mainly if you have only very few samples, we generated data sets that were smaller than our original data sets by deleting samples. So you can see for the first one we used a full data set, then we used only half of the samples, 1/3, and so on. These are results of a leave-one-out cross-validation. This means we always deleted one patient. We did the whole clustering, dimensionality reduction, and clustering procedure. And we compare the final clustering to the clustering of the full data set. So you can see as the size of the data set decreases, the robustness of the non-regularized version also decreases strongly. So we get a lot of variance in our leave-one-out clusterings, while for the regularized version, the results remain stable. Another question is if these clusters that we identified are also biologically meaningful, if they make sense. This is why we compared our clusterings to established molecular subtypes. Here's the result for the brain tumor data set, and there exist gene expression subtypes and DNA methylation subtypes. And you can see our 6 clusterings, and the first cluster is enriched in the mesenchymal subtype. Our second cluster is mainly a combination of the classical and the neural subtype, while our third and fourth cluster contain mainly proneural subtypes. So the question is, why didn't they end up together in one cluster? And the answer is actually here in the DNA methylation subtypes, because some of the patients are GSIM positive, while others are GSIM negative. So what we see here is that we use different input data types, and we can also see these input data types in the clusters that we identified. Finally, we had a look at how patients from different clusters respond to the same treatment. Again, this is for the brain tumor data set and a treatment with the drug temozolomide. So for every cluster, we generated a survival curve. So I just show you one in big here. On the x-axis, you see the time, and on the y-axis, you see the survival rate. And then we have the 2 curves, the blue one for the patients treated with the drug temozolomide, and the yellow one for the patients that were not treated with this drug. And what we try to figure out is to see if there's a significant difference between these 2 curves. So we did this for every cluster, and you can see here that in 3 clusters, it seems that the patients significantly benefit from the treatment with the drug, while in 3 other clusters, we couldn't detect any differences between the 2 curves. So this means that also from a clinical perspective, it might be interesting to look into these integrative cancer subtypes. To conclude, we introduced a method for robust integrative analysis, and especially it's also robust with very small sample sizes. Our identified cancer subtypes reflect information from different input data types, and they are biologically meaningful. We saw that concerning the survival times and also the response to the treatment. The method can be applied to data from various diseases, not only cancer. It can be used with different dimensionality reduction methods, also including semi-supervised ones, if one has labels for a small subset of the samples. And it can be used with different downstream analysis, not only clustering, of course. This work was done jointly with Nico Pfeiffer. I want to thank my home department at Max Planck Institute in Saarbrücken and all the people that supported us during this project, and the ECCB for the travel fellowship. Thank you.
Do we have any questions?
Thank you for the nice talk. I'm wondering, how do you find the number of clusters in your data set? I mean, because your survival analysis can be affected by number of clusters.
Yeah, that's true. So what we did, we used the silhouette value, which is a measure that for every sample identifies how well it fits into its own cluster and compares how well it would fit into the best other cluster. So for every sample, we get the silhouette score, and then we average over the whole data set. And this, by averaging, this gives us a hint on how dense our cluster are and how well separated they are from each other. So we actually cluster from 2 to 15 clusters. K means setting K from 2 to 15. And we choose that cluster having the best silhouette score.
And another quick question. So I see you are using TCGA data, but the number of samples you are using, they are quite small. For example, in breast cancer, it's around 100 something, while TCGA have a large number of samples. Why so?
So it's originally TCGA data, but we got this from the Similarity Network Fusion publication, and so it's not the most recent version of the data. So it might be from last year or so.
Thanks.
Yeah, I presume you're using some sort of linear kernels, not non-linear kernels, because effectively you're sort of doing like kernel PCA, and with non-linear kernels, you'd have some sort of problem with that.
Sorry, can you repeat the question?
I was asking about the kernels you use. Are they linear kernels or non-linear kernels?
We used Gaussian RBF kernel.
Yeah.
Yeah, so we used Gaussian RBF kernel in both scenarios. In the second one, we generated these 5 different kernel matrices by varying the kernel parameter.
Yeah.
But of course, one can use whichever kernel one wants and one thinks is appropriate for the data that we have.
Okay. How did you find the kernel parameter if you're using a nonlinear kernel, like a Gaussian kernel, and also the sort of number of dimensions you're going to use, which was your P?
We chose the kernel parameter according to a rule of thumb that was published by Smola and colleagues. So that takes into consideration the dimensionality of the data. And this is kind of the best guess that one can have about the data. And the number of dimensions that we project into, we did some analysis on that, and we tried between 5 and 15 dimensions, I think. No, we did between 2 and 15, and we saw that in a certain range, the results are relatively stable. One thing that we took into consideration is that we don't want a huge number of clusters, and in high dimensions, points tend to be far away from each other, and clusters tend to be dispersed. So we fixed that to 5 to avoid these problems with high dimensions.
So in the end, you're doing phenotype unspecific classification first, phenotype unspecific clustering, then based on that classification. Have you compared it with supervised learning methods?
No, similarity network fusion is,
For example, suppose that you'd like to do cross-validation on survival times or drug effectiveness.
We didn't compare to supervised learning because we actually wanted to do this exploratory analysis, and we wanted to see which clusters can we actually identify. And then the second step was this, this idea of seeing why are the clusters, why did they show up like they showed up. So we actually went the other way around without having this specific label. Okay.
Any more questions?
Thank you. I was wondering what will happen if you don't optimize the weight of your kernel. You just assign equal weights to each kernel, the result.
That's a very good question, and we didn't do that so far.
Okay.
Thank you.
Thank you.